Alrighty, scholars, we are back for part two, and I am going to be moving right away through this because my iPad is at 10% and we need to get through this. So, we have our main players. We're going to be using plasmid DNA, pathogen DNA, restriction enzymes, and ligase in this procedure. This next slide I'm going to title as our big goal. Okay. Ultimately, right, we know from our big goal up here, we're trying to create a subunit vaccine, so we need bacteria to produce pathogen proteins for us. So our mini goal out of that is to insert pathogen DNA into a bacteria cell. Now the question is, how do you do that? How do you get a bacteria cell to take in pathogen DNA? And what we use is kind of like the Trojan horse technique. We have to be able to sneak the pathogen DNA that we're desiring into a bacteria cell, and we do this using a plasmid because we know bacteria cells absorb plasmids relatively easily. So what we're going to do is we're going to try to sneak pathogen DNA into a bacteria cell by hiding it, by embedding it into plasmid DNA. So big picture wise, our goal, I can use the copy and paste tool I think here. Let's go copy. Oh, I did that wrong. Let's go duplicate. Okay, so our big goal is going to be to cut the plasmid once. We want to cut plasmid once, and at the same time, let me copy my pathogen DNA over here. At the same time, we want to cut the pathogen DNA twice. The reason we want to cut the pathogen DNA twice is we're trying to remove a gene from the pathogen DNA, right? The goal is ultimately to take out a gene for pathogen proteins. So what we do is we cut the pathogen DNA twice with the ultimate goal of taking that section that we cut out, taking that gene and inserting it into the plasmid. So where that one cut will be made in the plasmid, to open it up, we're going to try to insert the pathogen DNA. Now that brings us to our next question, which is, how are the cuts made? Okay. Remember, the cuts are going to be made by a restriction enzyme, and we know that what a restriction enzyme does is it cuts DNA at specific sequences. So we are going to choose a restriction enzyme that we know will only cut the plasmid DNA once and the pathogen DNA twice. What I'm going to do here is I'm going to zoom in on a section of my plasmid DNA. Dun, dun, dun. Okay, and DNA is double-stranded, so I'm going to draw the base pairing on the other side. Oopsie. A pairs with T, T pairs with A, C with G. Alrighty. So what restrictions enzymes do is they look for specific sequences of DNA and they make a cut there. Let's say, for example, that this specific restriction enzyme makes a cut anytime it spots the sequence TCGCT. That is the region that that restriction enzyme looks for and will always make a cut. So the restriction enzyme is going to make a cut at this location. Now, most restriction enzymes don't just make what we would call a blunt cut. It's not like it just chops straight across the DNA molecule. Rather, what the restriction enzyme is going to do is it enters in on one side of the molecule of DNA. It cuts through one strand. It's then going to cut up between the bases, and then it's going to exit on the other side of that strand. So it kind of makes this zigzag that it goes through. Now, why is this zigzag so important? And really quickly, I do want to draw in that this is my restriction enzyme that makes this cut. Why is that zigzag so important? Well, once this cut is made, what's going to happen is all my bases on the bottom part of that cut are going to be pulled away from the bases on the top part of that cut. Oopsie. So you can see now that once this has been treated with a restriction enzyme, oopsie, I now have two independent strands of DNA. And you should also notice, oops, I think I accidentally erased a letter in that process. You should also notice that on each side of where this cut was made, I now have 3 exposed bases that are not currently base pairing with any other molecules of, or with base pairing with any other nucleotides. What we call these exposed strands is sticky ends. Dun, dun, dun. Sticky ends. They're called sticky ends because these bases that are exposed are going to be trying to base pair with anything around them. Meaning, if they find a match, if this exposed sticky end of GCG finds CGC, it's going to base pair with it. It's sticky. It naturally wants to come back together. Now, why does this matter? Let's take a quick little zoom step out and look at what happens when we cut our plasmid DNA and our pathogen DNA. So in this next box, I am going to draw my plasmid DNA, but I'm going to draw it as though it's been cut open now. So I'm going to draw plasmid that has been cut open. But remember, it's not just that it was a straight cut to cut it open. Instead, there's going to be some exposed sticky ends. Just like how over here, when the cut was made, on one side I had an exposed GCG, and on the other side I had exposed CGC. I'm going to draw that in right here on this plasmid. So let me change the size of the pen here. On one side I have an exposed GCG, and on the other side I have an exposed CGC. And remember, those exposed ends, those are my sticky ends. Okay, so I have 2 sets of sticky ends right there. Now let's go ahead and also draw my pathogen DNA. So here's my pathogen DNA once it has been cut. So once my pathogen DNA has been cut on both ends, there should be sticky ends exposed on both ends, because let's look back at our pathogen DNA. When that restriction enzyme makes the cut, remember it's going to kind of cut it in this zigzag form there, and it's going to cut it in the same zigzag form over there. Now what this leads to is 2 exposed sticky ends. And on one side of the sticky end, there's going to be an exposed GCG. On the other side, there's going to be an exposed CGC. Once again, these are my exposed sticky ends. So what you should start to see now is that The sticky ends that have been formed on my pathogen DNA that I've cut out match up perfectly with the sticky ends that have been formed on my plasmid DNA that has been cut out. Okay, this end of the plasmid, base pairing wise, is looking for GCG. And so we have a perfect match between this end and that end. And then this side of the plasmid is looking for CGC. So once again, we have a match right there. So what's going to end up happening is I will have my plasmid. Dun, 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 dun. Let me redraw that. That is not looking too good. Okay. I will have my plasmid. And I had the sticky ends on both sides of my plasmid exposed. And then I also have my pathogen DNA now inserted into my plasmid, and the sticky ends matching up with each other in the plasmid. Okay, so now I have a whole completed plasmid, and now inside of this plasmid I also have a pathogen protein gene. Okay, and so now I've snuck the pathogen protein gene into my plasmid, and I can now give this plasmid to a bacteria cell. Now in tomorrow's video, I'm going to be going through what happens and how do we get the bacteria cell to absorb this plasmid. So we're going to talk about what steps are taken to get the bacteria cell to absorb the plasmid, and once the plasmid is absorbed, how are pathogen proteins going to be made and harvested to ultimately produce a subunit vaccine for us. So I will leave the video with kind of an overview shot of everything we've drawn so far, so you can screenshot that if you need to. And tomorrow we will talk about the next steps from here. Thank you for watching. Feel free to email me any questions you have. And remember, on Wednesday, Thursday, and Friday, I will have office hours to go over any of these concepts that might be confusing to you. Thank you guys. Have a good day.